um, pH, which means um, very low in the acidic level. By the way, this number here is only about 1.6 difference, but 1 point difference in pH means it's actually 10 times the acid level. So actually, 1.6 difference in pH is actually very large difference. So why do we study this? I mean, why do we study, what genes causes, G cell differentiation? By the way, when I say differentiation, it just means specialization. It's one of the terms that developmental biologists use all the time. I just want to make sure that you understand that. So why is, discovering NKX6.3 important? Well, it's important because it allows us to, know more about developmental biology, which again, I think that will allow us to really focus in on and give us more understanding of the cancer biology behind it. But what's important to know is that NKX6.3 is not the only gene that's important for G cell development. Okay, so there are at least 3 other genes that if you were to make mutant mouse of, you don't see G cells. So which means that NKX6.3 is very necessary. I mean, it's absolutely necessary to make G cells. But it's not the only gene that's necessary. There are many genes that are necessary. And we really do need to understand what happens with all of these genes. So for example, PDX1 or PAX6 or NGN3 that Dr. Leiter mentioned several times. How do they work together? And so, and what are the genes, sort of the combination of essential genes that's required to make G cells, or any other cell type for that matter? Well, so this leads to sort of the second part of the talk. Is this sort of simple diagram where we basically have this sort of almost like a ski slope where you have a ball. And you can actually go down different ski trails, okay? Now, the problem is that in the past, we used to think that once you go down a trail, you'll never be able to actually come back up or go sideways, just like in a normal trail when you ski. So for example, when you have a stem cell like this, the stem cell can actually take a phenotype or characters of pretty much any cell type in the body. So you can go down this pathway and become a stomach cell, okay? Or go down this pathway and become muscle cells. And this cell that has the potential to become, almost anything, as it goes down more and more, it loses its ability to become a specialized cell that's sort of outside that trail that you're sort of locked into. So we used to think that you cannot really go sideways or backwards. Well, this dogma has been now pretty much proven to be wrong. And in fact, you can artificially change the cell fate of these cells. And this leads to this slide, which is really the first paper that's published by Dr. Yamanaka in Japan that showed that even if you start out with a very differentiated or specialized cell like fibroblasts, which is a very common cell type that we all have, he can actually turn that cell into a stem cell just by making certain factors express in that cell. So what he did was that he actually figured out that SOX2, OCT4, KLF4, and c-MYC, which are 4 sort of genes that are important for stem cell. And so people knew that these genes were important for stem cells. They knew that they were expressed in stem cells and probably are important. But he actually figured out the right permutation, the right combination of these 4 genes, because there are many genes that are expressed in stem cells and probably important in stem cell development and maintenance. But he really figured out that these are the essential genes to make really pretty much any cells into stem cells, and therefore really making a groundbreaking discovery in stem cell biology and really cell biology in general. And he'll probably end up with a Nobel Prize for this discovery. So our lab is really at this time trying to figure out what are similar to what Dr. Yamanaka did, what are the set of genes that are important for differentiating or specializing and making different cell types within the GI tract, or for example, G cells. So we figured out, for example, through very basic biology, gene knockout in mouse, to figure out that NKX6.3 is one of those really important genes for G cells. But what are some of the other genes? And what are the minimum number of essential genes that are required and it's important for making these specialized cells, like G cells? So this is just a figure from our lab that really repeated some of the experiments that Dr. Yamanaka did. And for example, if you were to take these fibroblasts from mouse and express 4 genes, we were able to get a stem cell colony. This is not what fibroblasts, the starting cell type, look like. They never form these sort of colonies like this. And when you see this, it's a sort of a hallmark of a stem cell. Astonishing experiment. This was sent by Dr. Wilmot. He's the guy who actually made Dolly, the sheep who cloned one sheep to another. So he's another important scientist. So this is really I think the final slide. So Dr. Yamanaka figured out that fibroblasts, if you have these 4 factors, you get a stem cell. Another very distinguished scientist, Dr. Doug Melton, who's at Harvard, who studies insulin and beta cells that makes insulin and diabetes, therefore, figured out, for example, that these 3 genes, MAFA, PDX1, and NGN3, are the essential genes, basically, to make beta cells, at least in one experimental model. What about G cells? That's sort of considered the precursor of gastronoma. Well, I mean, there are many candidates like NKX6.3, PDX1, NGN3, PAX6, maybe others. Can we make G cells? What about cancer cells? I mean, can you actually make cancer cells into a different cell type? And I'm not in ways suggesting that we're going to have a cure with this sort of thing. But I think at the end, what's going to come out of it is that we're going to understand the biology of cancer better. So what are some of the factors that can actually change not just the normal cell type, but the cancer cell type? I think that's it. So anyway, I'll be happy to take your questions.
Terrific. Thank you very much. Yeah. What I'd like to do now is welcome the whole session 3 panel up to this little panel area, and I'd also like to invite Dr. Matthew Kulke from the Dana-Farber Cancer Center. We have some lingering clinical questions from this morning, and we're also going to throw those up to this panel. And if you guys could all please be sensitive to these microphones, you have to really get in there so that everyone can hear you in the back. And the first question that we have is, we all have talked a lot today about mouse models. So I'd really love to understand better, what is the role of a mouse model? How can we use these models to bring about better treatments and cures for patients?
Well, I think probably the best example is there are many types of malignancies where some of the initial screening for effective chemotherapy occurred in mice and in murine mouse models for leukemia and other solid tumors. Mouse tumors aren't always identical to human tumors, but they can be a good start if they're made in the right way. The reason for using the mouse is because this is an animal that is easy to manipulate genetically. For example, it would be much harder to do it in rats or primates or many other species. So that's the reason for doing it. And Ramesh, maybe you want to comment?
I'll only say that mice may not be small people, but people are big mice. The power of genetics is just uncontestable. It's very, very important. Mice reproduce from conception to delivery. It's 18 days. The women in the room are jealous. But it's 18 days, and then they're sexually mature by 6 weeks of age. So you can really cultivate mice very quickly and manipulate their genome pretty much at will.
And then Ramesh, how does understanding the genetic defects in mice translate into improved treatments?
So as I said, people are big mice. In other words, most of the genes that go into making us are present in the mouse. There's very little genetic difference, as it turns out, between mice and humans, or for that matter, between any mammalian species and humans. So really what makes a species distinct is very little tinkering at the edges of a substantially evolved genome. Once you have the power to genetically manipulate a species and understand the wiring diagram, so to speak, which has really been the emphasis of the talks in the last hour, now you can use the same animals to test treatments, to test genetic therapies, to isolate cells, to characterize the cells. It's just a very powerful model system to do the whole soup to nuts, except for the clinical trial. That has to happen in humans, just because even there, between monkeys or dogs or large animals and humans, there are substantial differences in metabolism and toxicity and the ability to deliver treatments.
One thing I'll add is sometimes the payoff in studying an experimental model is uncertain for years to come. In my talk, I talked a little bit about Wnt signaling, which is very important in a very common type of human cancer, colorectal cancer, and possibly in carcinoid. This pathway was originally discovered, described, and characterized in tail in fruit flies. And I don't think anybody ever imagined that it was going to have such an impact on human health. And there are clearly genetic forms of colon cancer that are directly related to mutations in this pathway. So it's very important having these genetically easy to manipulate model organisms.
Yeah, I think I just want to throw a word of caution into making too many conclusions from mouse models. There's absolutely no question that they're essential and a very important point for generating hypotheses, for making new discoveries. It's important, though, when you're thinking about a treatment, not to jump to conclusions based on experiments in mice. And many of you may be aware of a recent controversy in which in a mouse model it looked like angiogenesis inhibitors were in fact speeding up metastases, speeding up the tumors. That is something that absolutely has not panned out in human clinical trials. So one does need to definitely look at the human data in terms of making treatment decisions.
Thank you. What are the necessary steps to arrive at a cure for carcinoid and neuroendocrine tumors? Just, throw that out there. Anybody need some coffee?
Well, I think that is obviously a very big question, and I think really a lot of it is what we already just talked about. I think we have to really start from some of the observations from the bed, quote unquote, when we see patients, and these really incisive observations need to go, to create hypotheses. And because because we can't do certain experiments on people except, things like clinical trials, we need to have, obviously, a model organism or model that will allow us to answer some of those questions. And like what Matt just spoke about, I mean, it's really important to not jump to conclusion when we find even certain discoveries in mouse or any other organisms per se. But, again, this is just process of science. We need to, start with hypothesis, test it in models like mice. And then eventually when we have strong enough evidence, we can apply some of those, new knowledge or mouse knowledge to humans and see whether it holds up. But that rigorous process really needs to continue, and that's really still the nucleus of what needs to happen in order to cure any disease, I think.
And the rest of the panel?
More research. Plain and simple.
I think my view is it's very important to have the physicians and the scientists talking to each other. I think it's very important for the physicians not only to understand the science as it comes out, but to communicate their view of the clinical problem to the scientists, many of the scientists being physicians themselves, as many members of this panel. But I think by the same token, it's very important for the scientists to communicate effectively with the clinicians. I think it's important for us to make our clinical colleagues aware of where what the discoveries are illustrating, to work together to devise creative clinical trials that are going to push the limits, not simply extend life on average by a few weeks in a large cohort of patients, but to really be a game changer. I think we need just ongoing, continued mutual respect and interaction among people from different disciplines.
So I think I would reinforce that. It's really been quite amazing just over the past 5 years to see on the clinical side the development of a wealth of new treatments that are so-called targeted therapies that really were developed and can take advantage of biological understanding of tumors. So we've seen huge progress there. In parallel, we've seen huge progress in genomics, as I think we'll hear about later, and in basic biology in mouse models. And I think there's really an opportunity in the next few years to see a confluence of all of those developments and really make some dramatic progress in this disease.
Thank you. The final question for the researchers is, how can this research into carcinoid and neuroendocrine tumors be applied to other diseases such as diabetes and also other cancers?
Well, one of the, I think, bright sides to what's going on in research in this field studying enteroendocrine cells is the realization in the last 5 to 10 years that they are very important for a lot of common clinical conditions in addition to somewhat uncommon neuroendocrine tumors. So within the last 10 years, there's come to be an appreciation that one of the major targets of the gastrointestinal hormones is the brain, particularly the parts of the brain that control food intake, how much you eat, how hungry you get. And as you know, obesity is a rampant public health problem in the United States. Likewise, the pancreatic beta cell that makes insulin is a major target of enteroendocrine cells. And the discovery that you can actually improve insulin secretion with gut hormones really means that this is another very common disease affecting 10, 20 million people that is potentially impacted by research on neuroendocrine cells in the GI tract. So I think that it's going to attract a lot more good scientists to the field, which is, I think, good for people looking to see progress in carcinoid.
I think it's also worth pointing out that the reverse is probably equally true. It wouldn't surprise me one bit if the cure for carcinoid came not out of this community, but from people studying diabetes or people studying brain hormones. You know, one of the most attractive things to me in science is the continuous element of surprise. Andy made a very important observation earlier about how our cardinal understanding of colon cancer genetics originated from research in fruit flies. And, you know, who'd have thunk that? So I think the important thing is for scientists to have an open mind, to be aware about what's happening in other fields, and to continue to exercise creativity at the very edges of human understanding.
Thank you. I'd actually like to go back. Dr. Kolke, you brought up earlier the importance of bringing about targeted therapies for patients, and some of your colleagues this morning touched upon the importance of phase 2 and phase 3 trials in those patient populations. And so we have a large group of patients here. What's necessary to bring about those types of results and to get the answers that we need to bring about these targeted therapies? What would it take to complete these large phase 2 and phase 3 studies?
It's been interesting. So in the sort of late stages of drug development, one needs to do what's called a phase 2 study, which is just seeing if the drug works, and then to really make a drug widely available for patients, very often one needs a randomized study in which there's a comparison between how patients do who receive a drug, either to placebo or sometimes to another drug. There had been a lot of concern because the impression had been that this is a very rare disease, that it was impossible to do trials like this. What's been amazing is that in many cases, these trials are getting done more rapidly now in neuroendocrine